Hi everyone, my name is Murat. I'm the CEO and co-founder of Antiverse. Today joining me Tzvika Hartman, SVP of Computation from Biologic Design. We are very happy to have you here, Tzvika. Welcome.
Thank you very much for the kind invitation. I'm really excited to be here.
I'll just dive in. Most of the people who are on this call already know quite a lot about you, but would you mind mentioning us the defining moments in your life that brought you to this role at Biologic Design?
Yes, sure. We'll be happy to. So my background is actually started in mathematics and computer science. Did my graduate studies in the Weizmann Institute. On theoretical computer science. In my PhD, I also did some work on algorithms in molecular biology and DNA arrays. Back then, genome rearrangement, but more on the algorithmic side. And after 2 years of postdoc, actually was in the joined the early days of the Google office in Tel Aviv. Started working on the search engine, quality, nothing related to biology. Then also some other various projects as a researcher and a software engineer in Google Trends and infrastructure and so on. At some point after 7 years, something like that, I wanted to do something which I felt is more meaningful. So I basically initiated some project around health tech, got some pitched to the executives and got actually the opportunity to build a team, which eventually grew up into the Google Health team in Tel Aviv. I managed this group. We did some machine learning to help physicians get good diagnosis and decisions and so on. And after a few years, I actually left Google. I thought that maybe a better opportunity will be to contribute to the biological world with my computational experience and expertise. And I joined Biologic Design. I'm 2.5 years at Biologic. I'm leading the computational department. So that's what got me here. Very excited in my work.
Definitely so. It's such an exciting moment for companies like ours working in the intersection of biology, AI, drug discovery. Especially on the biologic sides, there wasn't too many players, but Biologic Design is one of the earliest ones out there. What excites you at work? What's the the most interesting projects you guys are working on now?
So at Biologic Design, we actually try to make what we call smart antibodies. So antibodies, therapeutics are around for some dozens of years, but most of the antibodies are basically act as the small molecules, but as an antibody. They agonize. They don't actually exploit the potential of proteins, which are basically nanorobots that can do very special things, respond to the environment, act as do some basically sophisticated tasks in our body. So what we do is using AI to basically program the antibodies to do things that we want them to do, which are quite sophisticated. So basically, we have two main directions. One is epitope specific, which we by the AI we can design antibodies that will bind to a specific epitope in some antigen. Actually, there is now in phase 1 in the clinic. We have our first antibody that is epitope specific. It's called AW07, and the results of the clinical trials are quite promising. This is one direction, as I said. The other direction is what we call a multibody. This is an antibody that can bind in the same arm two different antigens, which it's a very powerful mechanism. What we're doing is using AI both in designing the libraries that will optimize the probability that we will get good candidates, and also at the end of after screening the libraries, we have a pipeline that gets the results from the lab, the NGS, all the measurement that we've done, and we basically train models based on these data sets, on this data, and by these models we can predict in silico essential properties of the antibodies like affinities. We can build a model for predicting affinity of the first antigen in multibody, for example, predict affinity for the second antigen, predict developability properties, stability, etc. And all these suite of computational tools and lab expertise allows us to develop antibodies which can do what we call nanorobots, which can do different things than regular antibodies.
Yes, that's great. When you mentioned about this multi-specificity in a single arm, that's such a, I think, novel concept. Maybe I heard it once before in another company, but what's the advantage of? I think a lot of people right now, when they do the bispecifics, they design two arms that binds to two different antigens. But you are basically saying that one arm binding to two different antigens and having a symmetric antibody, right?
Yes.
So what would you say the advantage of your approach over bispecifics?
Okay, so first, one of the advantages is that in the half-life and developability aspects of the antibody, because this basically is a natural antibody. A bispecific is not a natural antibody; it's an engineered antibody. And therefore, some properties may be less favorable for antibodies. What we are doing is a totally human antibodies with few mutations, but everything, all the infrastructure, I'm using a computer science term, of the antibody is the same. So this is gets the risk is much less in the function of the antibody. This is one thing. Another thing is that it can allow us to do some like smart things that you cannot do with bispecific. I'm not sure I can describe exactly, but these properties. But the fact that the same arm can bind different things, you can look at it as kind of some AND gate or gate in terms of Boolean computations, which allows us to do some special computations and act differently in different tissues and different microenvironments. Depends, for example, on the concentration of one antigen. You can do some other things with the second antigen. So this concept of multibody opens a whole suite of possibilities that you can basically program the antibody to do some things that cannot be done by other strategies, by other technologies.
Yes, that and or type of logic is also almost embedded in the name of your company as well. And I know that it was a concept that a lot of CAR T companies struggled with. So, to activate killing only in the presence of two antigens, especially, for lots of oncology. So that's great. Very helpful. Thank you very much. So that we might have some people in the audience never used the AirMeet before. If you look at the messages section on the top right, there are, and click on that, there are 4 little icons on the left. One of them says Q&A. If you have any questions, please write down there, and we will take it at the end of the session. We are planning to continue this conversation for another maybe 15, 20 minutes. Take a couple of questions, and then we will break out to a networking session area, completely optional, for about up to 90 minutes. Great. So what you described already, very, very exciting platform technology that might apply in so many different areas. When you think about the future projects that you might be undertaking, can you tell us a little bit more? What would be the new type of therapeutics or the new type of targets that you could go after with what you are building?
Yes. So I think that, as I said, the technology that we have opens a whole world of possibilities. I can give maybe one example. One of the maybe even two examples. I'll start with one of the things that we developed. So this is basically an antibody that is for autoimmune diseases, and it can bind. It's a multibody that can bind both IL-13 and the TSLP, which are both targets, validated targets for autoimmune diseases such as atopic dermatitis. And the fact that basically there is antibodies for each of the targets separately, but in the data out there suggests that there is a limited effect of each of these targets, because if each of these drugs, of these antibodies, because, for example, if there is a patient or a tissue where most of the I won't go into the biologics, but there is a drug, tezepelumab, against TSLP, but in a microenvironment where there is not much TSLP, there are other cytokines, then this drug won't be effective, and vice versa. In a microenvironment where there is IL-13, not enough IL-13, the other drug won't work. And the fact that we have, it's 2-in-1, we have one antibody that can chase basically these two cytokines, these two targets, is kind of like, in a very metaphorical way, it's kind of precision medicine. I mean, you don't need to you can give it to like one size fits all. You can give it to a patient and it will find both the IL-13 and the TSLP. Another concept is what we call super engagers. Is something that is like there is the T-cell engagers, which there are some drugs for this. But here in each arm of the bispecific super engager, we put a multibody. So now we have, again, if there is some TAA that is targeted by some engager, then it will target only tissues where this TAA is present. But if we target two different TAAs, the probability of success, basically the percent of cases in which this will work is much higher, because we have, again, one size fits all. So this is something that this concept can be used in very creative ways. So this is some examples about things that can be done down the road.
Yeah, I can definitely see the value of what you're building in those particular scenarios, and a few more. Yeah. I have a few more questions, but I'm also checking the Q&A channel to see if we have something that might be more context sensitive. So there is one, if you don't mind. They ask, Zeta from King's College, how do you design your antibodies for properties like ADCC, depending on, obviously, the epitope that antibody is designed for? Let me show it on the stage.
Not sure I understand. the question?
Maybe they are just asking which parts of the antibody you have to adjust for ADCC, but we'll bring maybe Zeta later to the network question and answer it.
Yes.
You just pull that up. So there is another one, and this is about the data you guys are working on. Is it mostly public, or is it the data that you generated in-house?
So both. We are basically pre-training our models on public data, proteins, antibodies, etc. But the game changer is the data that we are generating in-house, because what we generate in-house is data for the affinity or other properties of the specific antigens that we're looking for. So the short answer is that we use both. We build on pre-trained models and data on public data, but the game changer is by using our data, proprietary data that we generate in our lab.
Okay, thank you. There might be some follow-up questions on that one, but better to get into those, I think, in the networking session. So another one, let me rephrase this question. They are asking about the agonism. So I know that, a lot of companies are going after binding. Some of them are antagonism. And agonism seems like an area that is avoiding a lot of people and quite desired by many. Do you have any examples of designing agonistic antibodies against, membrane proteins, for
example? So we have an example that our antibody that is in the clinic is designed for IL-2. Basically, IL-2 is of course used as a drug for cancer, for example, but it's a double-edged sword because it can help the immune system and activate it and increase it, but in some cases it can also decrease the immune system. So this is also can be very toxic. What we developed is an epitope-specific antibody that binds the IL-2 in such a way that it will prevent it to decrease the immune system and only will increase the immune system. So this is an example of a way that we basically activate the molecule, not prevent it work, but activate it. And I want also to maybe this is also an opportunity to mention is that the biology of this is validated and well known for many years, but till now no one knew how to develop the ammunition to tackle this. So this is an example of a validated biology that is well known, that our technology allowed us to engineer the specific solution for this problem.
So amazing. So I will move on to another question. When you think about the future of drug discovery, I'm sure you have faced your fair share of the skeptics when it comes to application of AI. And rightfully so, I would say, given the previous experience people had. Do you think that this time is different, combined with AI, when you look 3 years, 5 years into the future? How do you see the landscape changing?
I think there's a lot of potential here, which is not yet unleashed. First of all, we can see the incredible advances in AI, in NLP, and ChatGPT, all these buzzwords. that really do amazing things in natural language that no one, I don't know, maybe not no one, but I think not a lot of people have predicted that it will happen so fast and so good. And I think that these incredible progress can also be done in the biologic language. Of course, this is much more complicated. I think we don't have the same intuition that we have into natural languages because, it's a language that the protein language, the DNA language is of course something that we as humans have less intuition into it, but the data is there. And I think that the combination of the computational progress and the biologic expertise will be the game changer. And I think when we discussed it before the meeting, that the secret sauce here is how can we integrate between these two disciplines. I think this is something that everybody talks about it, but I think it's really hard. I mean, it's really hard to get people that are experts in one of the disciplines. For example, people that came from the computational world and did very deep and have deep understanding in it, and on the other side, people that have a lot of experiences and expertise in biology and experiments, and getting these two experts to the table and have really a deep, meaningful, fruitful discussion is something that is not trivial at all. But I think this is the key for success for this area. I think this is the main barrier now.
Yeah, proficient. I always like to say we need people who are likewise, proficient in Python and pipetting same level. And there are only a few of them, only a few unicorns, people say. But obviously, there's a huge talent gap there. A lot of people see it. I think it will be filled either by companies cross-training people or new university programs. Maybe pharma having their own programs to 6 months, 12 months train people before they, start their actual work. So there are a few options there. Talent aside, what would you be looking for from the audience?
So I'll talk first about some general things, I think, for the industry. So I think more public data will help everyone. Probably there is a lot of data, especially in big pharma, of different sequences, variants with data, valuable data of affinity, developability, stability, and so on. So sharing more data, I think, will be in the benefit of everybody. This is one thing. Another challenge, which it's something that we talked about also now, is how can we get people from the tech industry to go into the biotech industry. There's obviously a lot of talent out there, and I think that unfortunately most of the talent is directed toward different high-tech things, internet, cyber, you name it, and not enough to the biotech. So we should think about how can we attract more talent to this such an important discipline. And also you talked about it before, how can we train people to be experts in both disciplines, which again is something that we should think about as a community. Speaking specifically about biologic design, so our main business is developing our own assets, a pipeline of drugs that we are bringing into the clinic. But we have also another aspect of collaboration with big pharma. We have collaborated with we had successful collaborations with Novartis, Eli Lilly, and some other companies, AstraZeneca. And we are basically looking also for opportunities where we can collaborate with big pharma and in projects in which our AI and other innovative technology can give an added value. So this is also something that I would like to talk to call the audience about.
And on that collaboration point, would you say that it will be the pharma coming with their own targets and then applying your platform and technology to come up with smart bodies against that?
Yeah, this is one possibility. The other possibilities is that we have discussions and back and forth about the needs of the big pharma and which technologies we can use. Which of the technologies that we can use can help them, and this is kind of a dialogue between the big pharma and us to find the exact target and drug which we can have the added value, and it aligns with the business considerations of the pharma.
Thank you. I'll quickly screen through the questions we have. There's one question on a very high level. When you are describing Biologic Design, do you guys think yourselves as a tech company, a biotech company, now the new term Techbio company?
Yeah, so I think it's more like I don't know the term Techbio. But what is Techbio?
I believe it's the application of a technology as a platform to a topic of biology, but tech being first or having more emphasis than bio.
Yeah. So I think, we're not a platform because we have our assets and we develop them. But I think that it's more accurate to describe us as a tech company rather than a bio company, because we are taking validated biological targets and the added value that bring is the technology. So the risk is like we take the risk in the area where we are experts. The risk is in the technology which we feel like we are the experts, so we're willing to take the risk. So basically, I think we are more a tech company than a biotech company.
Thank you. I see there are a few more questions, but it might be more suitable to take them on the networking session. On behalf of everyone in our audience, we would like to thank you, Tzvika, for joining us and having a great conversation. And on that note, we will slowly move on to the breakout room. And people, if you haven't used AirMeet before, there will be a couple of virtual tables. There will be some representatives of Biologic Design at different tables. You can just go and sit on those tables and have a conversation for another 60 to 90 minutes. Thank you all. Thank you, Tzvika.
Thank you very much, Murat. It was a pleasure.
Likewise. Take care now.